Hi everyone, we're going to continue on with our cell organelle lecture, and this is part 2. You may remember that we left off with some information about mitochondria and chloroplasts. So we kind of differentiated between the two. We learned that mitochondria was in both plants and animals, and mitochondria make ATP through a process called cell respiration. And then we learned that chloroplasts are found in plants, and chloroplasts use the process of photosynthesis to make sugar. However, one question we didn't answer, and an important piece of information that you do need to know, is about the endosymbiotic theory, or the endosymbiosis theory. It was come up in the 1980s by Lynn Margulies. This theory tells us about the origins of mitochondria and chloroplasts. So the theory basically tells us that these two specific organelles were once free-living bacteria. They were prokaryotes. And they got engulfed by an ancestral eukaryote. So an early eukaryote, not quite the eukaryotes we know today, but a eukaryote very early on. And when they became engulfed, they existed as an endosymbiont. So endo means like inside, and symbiont or symbiosis means living together. So we have one thing living in another thing, a cell that lives within another host cell. This would have happened as a partnership, and there would have been an evolutionary advantage for both. One supplied energy, and the other supplied raw materials and protection. So the theory basically goes like this. We have an ancestral prokaryote, and over time, this ancestral prokaryote might have become larger. And as it became larger, some of the membrane infolded for some reason. There may have been some mutation or some reason why some of that cell membrane might have folded in on itself. And part of that folding in could have formed a little barrier between the rest of the cell and the genetic material found in the middle. This might have been an early nucleus. Now, why would this have been helpful? Can we think of any reasons why that barrier might be helpful? Well, it could be due to protection, right? The infolding could have helped protect the nuclear material from damage. Over time, the infolding could have grown more complex and formed structures that we now call the endoplasmic reticulum and the Golgi apparatus. So this infolding happened, and basically now we have an early eukaryote. So now we have these early eukaryotes. They have some advantages. That folding allows for protection of the nucleus. Some of these early eukaryotes may have engulfed what's called down here a heterotrophic prokaryote, a small prokaryote that entered the cell. So see how it was brought in, and now that prokaryote is living inside of the eukaryote. This is the endosymbiont. So think about it. The prokaryote is making ATP. That's its job. Its job is to make ATP. It makes lots of ATP. So because the prokaryote is making ATP, the bigger cell also benefits by getting ATP. What is the bigger cell doing that benefits the smaller cell? Well, the bigger cell is providing protection. It's providing some raw materials for this prokaryote to grow and divide. So we think that this is how the mitochondria came into existence, that they were engulfed, and we had this mutual beneficial relationship, the mitochondrion producing ATP, the rest of the cell providing protection and raw materials, and now we have eukaryotes with mitochondria. Then the theory continues that some of these cells that now have mitochondria may have engulfed another prokaryote, and this prokaryote was photosynthetic, which means it took in sunlight and carbon dioxide and produced sugar. So this, again, would have been mutually beneficial. Producing sugar would have benefited the bigger cell, and the smaller cell would have benefited from the protection and raw materials. So now we have an early chloroplast. So the theory goes, that is how we have these two organelles. The mitochondria has to be first because every eukaryotic cell pretty much has mitochondria, but not every eukaryotic cell has a chloroplast. So a chloroplast may have come later. So that is the endosymbiotic theory. So some questions for you to answer. What are the steps of the endosymbiotic theory for a plant cell? Question 2, what is different? And question 3, which evolved first? So take a few moments and answer those questions. We just answered those a few seconds ago. Okay, so this is a theory, right? And if it's a theory, then we need evidence. What's the evidence to back up the theory? Well, there is quite a bit of evidence, and it's outlined in your book. We're going to go over some major points here. Mitochondria and chloroplasts have their own circular DNA. Now, that's important because our DNA, eukaryotic DNA, is linear. It is not circular. So what has circular DNA? Bacteria have circular DNA. So that's a big piece of evidence. Mitochondria and chloroplasts have their own DNA, which is already kind of weird for an organelle to have its own DNA, and it's very similar to bacterial DNA. So that's a big hint that they used to be bacteria. Mitochondria and chloroplasts divide via binary fission, and that's the same way that bacteria and prokaryotes divide. So they have similar cell division as bacteria. They have double membranes, just as they would if they had been engulfed. Their innermost membrane is similar to bacteria, and their outermost membrane is more similar to eukaryotes. Furthermore, the ribosomes in chloroplasts and mitochondria are more similar to bacteria than they are to our own cells. So they even have their own ribosomes. And those are some major pieces of evidence that tell us that this theory is probably right on. So it's a largely accepted theory that this is how mitochondria and chloroplasts came about. Mitochondria are found in plant and animal cells. You see them there in animal cells and plant cells. Chloroplasts are only found in plant cells. Okay, so there we see some of the things we've been talking about: lysosomes, cytoplasm, vacuoles, mitochondria, chloroplasts. All right, so that is how we get energy. We use the membrane, we use vacuoles and vesicles, and we use the mitochondria and chloroplasts. What else do our cells need to do? Well, one important thing is that they need to make proteins. Proteins help to run our daily life. The cell needs to, in order to make proteins for our cells to do everything they need to do, we first have to read DNA. Then we have to build the proteins. Some proteins are structural, like our muscles are made of proteins, our hair and our skin. If you're an animal with claws, that's made of protein. Proteins are also enzymes. Enzymes speed up chemical reactions. They're very important. Proteins are also signals, so they help our cells communicate. So what organelles do we need to build proteins? We need nuclei, we need ribosomes, we need ER, the endoplasmic reticulum. We need a Golgi apparatus. Okay, so proteins do all the work. One of the major jobs of cells is to make proteins because proteins do everything in our bodies. They do a lot of work. DNA is the code for the proteins of our bodies. All of our proteins are encoded for by DNA. So DNA is the code for proteins, and proteins do all the work in our cells. So a very, very important job. And there are some of the jobs that a protein will do in our cells. We start in the nucleus. The nucleus is our cell's control center, and it is protecting and holding all of our DNA. Our DNA is very important. Your DNA is simply instructions for building proteins. So it's found, if this is our nucleus, the DNA is found all throughout here. The structure of a nucleus is we have a nuclear membrane. Notice it's packed with all these holes. Those holes are so certain things can get in and out. Those are called nuclear pores. The nuclear membrane is sometimes called the nuclear envelope, but it's the same thing. Inside of the nucleus is something called the nucleolus. It's a little dark spot in the nucleus. That's a nucleolus. What does the nucleolus do? The nucleolus builds another structure called ribosomes. We also have chromosomes. Our DNA is organized into chromosomes. So that is also found all throughout our nucleus. All right, so that's what we've got first, nucleus. So there it is being outlined in red, protecting the DNA and controlling the cell. Now, in addition to our nucleus, we need ribosomes. Ribosomes are very tiny, and they're not actually membrane bound. Ribosomes are so important that they're found in all cells, prokaryotes and eukaryotes alike. Ribosomes are protein factories. They basically read instructions and they build proteins. Some of the ribosomes are floating around free in the cytoplasm, and some are attached to the endoplasmic reticulum. So we see them here on the ER. So here are the free ribosomes. The free ribosomes are just floating around. These free ribosomes are reading something labeled as mRNA. mRNA is like a copy of DNA. So DNA gets copied into mRNA, and mRNA meets up with a ribosome and makes a protein. Ribosomes that are freely floating in the cytoplasm make proteins that are needed in the nucleus, that are needed in the mitochondria, that are needed in chloroplasts, or that are just needed in the cytoplasm. This is something that you should remember. So make sure you highlight this, circle this. What kind of proteins do free ribosomes make? It's listed right here at the bottom. Some ribosomes are situated on the endoplasmic reticulum, the ER. So again, here's a ribosome working the same way. It's reading the mRNA, but it's feeding the protein. Notice it's feeding the protein into the endoplasmic reticulum. So this is going to do something different. These proteins are going to go to the plasma membrane, or they're going to go to a vesicle and they're going to leave the cell, or they're going to become some of those enzymes that are found in lysosomes. Remember that lysosomes contain digestive enzymes. So, and highlighted in red are all of our ribosomes. Some of them are free, they're just floating around the cytoplasm, and some of them are found on the rough ER. All right, then we need the endoplasmic reticulum. The endoplasmic reticulum is a series of membrane-filled sacs that work on proteins. They basically help to finish the protein's shape after the ribosome builds them. So remember how I said that proteins have a very specific shape? Well, the rough ER is going to help work on that shape. The rough ER is also going to make membranes that protect the proteins that it's making and working on. So the rough ER is going to have ribosomes attached to it, and the rough ER is going to work on proteins. There is another type of ER called the smooth ER, and the smooth ER has nothing to do with proteins. It's got a totally separate function. Its function is to make membranes, to make lipids, and to make detoxifying enzymes. So highlighted in red is the ER. The stuff with the ribosomes is the rough ER. The stuff without the ribosomes is the smooth ER. All right, so in addition to the rough and smooth ER, we also have the Golgi apparatus. The Golgi apparatus's function is to modify, sort, label, and ship proteins. It's sort of like UPS. Basically, the proteins arrive at the Golgi apparatus. The Golgi apparatus is the shipping and receiving. The Golgi apparatus decides, well, this protein needs to go to this cell. This protein needs to go to another cell. So the Golgi ships proteins in vesicles, like UPS trucks, right? And the UPS trucks send it where it needs to go. The vesicles will be sent where they need to go. So the Golgi, kind of like the rough ER, looks like a stack of membrane sacs. All right, and there is the Golgi. You can see it looks similar to the rough ER, but it has its own distinct shape. All right, so let's review some of the organelles that we've learned so far today. First of all, we have the nucleus, and in the nucleus we have our DNA. The DNA is going to copy itself into mRNA, and that RNA is going to leave the nucleus through one of those little openings, the nuclear pores. Some of the mRNA is going to meet up with ribosomes that are attached to the endoplasmic reticulum, the rough endoplasmic reticulum. There is an example of it happening. And what's going to happen there is the mRNA is going to meet with the ribosome, and it's going to make a protein. The protein is going to move throughout the rough ER, and it's going to get folded appropriately. Right, you see that happening right there. All right, now a vesicle is going to bud off the rough ER, and it's going to meet up with the Golgi apparatus. The Golgi apparatus is going to package it and ship it and put on a tag that sends it to the right place. And then it's going to be sent in a vesicle out of the cell. Notice that this protein is going into the blood, and it's going to travel somewhere else in the body. And that is what happens to make a protein. All right, nucleus, nucleolus, ribosomes, the rough and smooth ER, as well as the Golgi. All right, and finally, we're almost done. The last thing that cells need to do is to make more cells. So we have a whole chapter on this, but basically, in order to make more cells, the cell really needs to grow. Sometimes the cell needs to repair itself. The cell needs to replace broken or damaged parts. So in order to do this, the cell has to copy its DNA. It's got to make extra organelles. And then it has to divide the DNA and the organelles in half. And then it needs to split in two, as you see in this picture. We have two cells coming from one. The organelles that do that work are the nucleus and centrioles. And we'll get to this more later. Here are the centrioles. The centrioles are helping to divide everything in half. All right, so there's that. Our last question, what kinds of proteins are produced by cytoplasmic ribosomes? You may need to look back in your notes. Those are free ribosomes found in the cytoplasm. What kinds of proteins are produced? Put your answer in. And you should remember that the proteins produced by cytoplasmic ribosomes are freely floating in the cytoplasm, or they may be needed in the nucleus or the mitochondria or chloroplasts, or as part of peroxisomes. All right, that's all we have for today, and that is our overview of organelles done. If you have any questions, please let me know.